Hello, everybody, and welcome into our Physics and Astronomy lecture for the month of May. Before we get into today's lecture, I did want to give a couple of updates on our department. I did want to extend a couple of congratulations to SWOCC physics and engineering students, Troy Liggett and Ethan Killinger, who both completed their NASA projects for this year and will be presenting at the Oregon Space Grant Consortium symposium next week. I also do want to take a few minutes to pay tribute to three people that have passed since last we had one of these, that have either played a role in my personal career or in the development of our department. One of which being Dr. Reginald Dufour, who actually was a Bandon resident and one of my former instructors at Rice University when I was in grad school, who recently passed a few weeks ago. We also had one of our SWOCC students who was part of my SPEAR team, the balloon launch team, a welding student by the name of Matt Thomas, who unfortunately passed away this week. And I was also informed yesterday of a friend of the department, Ms. Kendra Perry, who was responsible for me getting opportunities to participate on Hooked on Oregon and a number of other media outlets to promote the department, had recently passed due to complications of some medical procedures that she was undergoing. So today's lecture is kind of in tribute to each of those three individuals for their contributions to our department and to the science that goes on here at SWOCC. I am proud to introduce sort of a homegrown talent for this round. One of our wonderful nursing faculty here at SWOCC, Zinzi Konig, formerly a emergency room nurse at Bay Area Hospital, and before that a research scientist at University of Texas Medical Branch, I believe in lovely Galveston, which is a very nice place to study. She will be presenting on some of her research and some of the research that has gone on in terms of the effects of spaceflight on human health. And with that, I will turn things over to Zinzi, and everyone enjoy the presentation. I'll be monitoring the chat. If there are any questions, we'll have a Q&A afterwards, and I will pass those questions along at the end.
Hello, everyone. Thank you for joining in to this talk about the effects of spaceflight on not only human health, but also the infectious agents that we might carry along with us in our journey to space, and that we do carry along with us when astronauts board the shuttle, or now the Soyuz capsule, and reside on the ISS, or take missions back and forth. The reentry forces, the gravitational sensing mechanisms within cells, and the alterations in gene expression are really what I'll talk more about today. So it's a highly technical lecture, but I think it should be enjoyable. Here in the corner is the mission symbol or mission patch for the first flight experiment I'll talk about on Space Shuttle Atlantis. It was called STS-129, or Space Transport System 129. There will be lots of acronyms today because we are talking about NASA projects and spaceflight. Nearly all of what I'm going to present today is available to anyone, has been made available to anyone by NASA, the Translational Cell and Animal Research in Space that took place from 1965 until 2011. And so this is encompassing the data that was generated in experiments involving humans and animals and plants, living organisms, in space from the Mercury era, so very early on, 1965, all the way until the end of the shuttle era, where my last experiments were in 2011. So we're going far back in time with much of this data, and NASA Ames, their Ames Research Center, really made this document available because many of the findings of flight experiments are they are a single experiment where there is no real ability to replicate microgravity, right, on space on Earth. And so reproducible results are not necessarily the objective. The objective is to gain knowledge and then put all of that together in sort of generating countermeasures and additional hypotheses and additional questions to ask for the next generation of people that will take over the space program and be investigators. And so that's why this talk is so important to give to a group of young students and educators. And so this is the document. It was compiled. My old mentors all contributed to it. Some do have publications. We're able to do ground-based modeling experiments, but anyone can access this information. It's made freely available by NASA. And so my role in the space program was really, it was translational from a distance. I started out as a registered nurse, working in a variety of areas. And when I had gotten significant experience in that, I entered into a master's program in public health, really actually global health, specifically in the epidemiology of microbial diseases. So viruses, bacterium, parasites, all infectious agents actually, including even prions, which are protein-based infectious agents. And so studying the transmissibility, the communicability, and the epidemiology of these agents is what public health or epidemiologists concern themselves with. And so even as an early nurse, I was able to understand what a pandemic was, what an epidemic was, and how the countermeasures that we have in place, specifically levels of biosafety, particularly in a clinical environment, since that was what I was used to, and what that really means in spaceflight. And so I generated these hypotheses on my own at the end of my master's program in epidemiology and global health. And after my experience in working in a microbiology laboratory and looking at gene expression, alterations in gene expression, and how that affects not only virulence, so the severity of disease, but transmissibility between individuals, and the mechanisms in which that transmissibility or virulence changes. And at the time, I was specifically looking to tease that out between infectious agents that are transmitted via a droplet route or versus an airborne route. So an airborne infectious agent, right? We have more global knowledge now about this thanks to the pandemic, but droplet precautions were usually implemented in patients or in laboratories, certain biosafety levels, with the prediction that there would be no transmission or communicability of the agent without a very without a mask that is not an N95 mask, which is just a surgical mask. And so we now understand there's a difference, right? The nanometers or space between the holes in the mask and how much particle, how small a particle can get through. And so it was really believed, and it was the dogma at the time, that there was only 3 airborne infectious agents, and that is tuberculosis, measles, and varicella zoster virus. Measles and varicella zoster Measles is controlled with a vaccine, and so it's not something we typically worry about in this country. Tuberculosis primarily affects underserved groups or populations that cannot afford new medications. And so the concern of over tuberculosis being airborne and droplet was established. It should be 1 to 2 meters. That held in place with measles and with varicella zoster virus, the virus that causes chickenpox. All can be considered airborne at some point in infection. And other infectious agents, like potentially Streptococcus or a meningococcal meningitis, the precaution is different. It is not something that can be transmitted in an airborne route through ventilators or become attached to a particle of dust suspended in the air because it is so light, the way measles can. I had a specific hypothesis that microgravity will alter the transmissibility of droplet and airborne infectious agents by separate mechanisms within the host and the infectious agent. But what I was really concerning myself with was the 1 to 2 meters, the dogma of epidemiology that infectious agents can be partitioned into droplet infection and droplet-based communicability, like a cough, cold. Those are droplets in the air that eventually, because of gravity and their weight, these infectious agents fall to the ground and are not then traveled through an airborne route. I did many of my own very rudimentary experiments with this 1 to 2 meters. I really tried to distinguish where this is based off of and how in a contained spaceflight vehicle. And so back to my role as an epidemiologist and concerned with global health. In an instance where long-term habitation in space is necessary for a large group of a diverse population that is not necessarily the right stuff or gone through continuous screenings as an astronaut does, whether or not in that contained vehicle, perhaps a generation ship, an agent that is formally thought of as only transmissible through droplet method. So somebody has a infectious disease that normally should not travel beyond the 2 meters in order to be caught by another host. I had the hypothesis that microgravity would change this, and that this 1 to 2 meters does not apply in space. And so as a future thinking epidemiologist, I concerned myself with the potential habitants on a contained spacecraft in microgravity and the infectious agents that we would bring with us. And so in order to do this, I had to find where investigators were looking at these questions already, similar questions. We, of course, need to investigate and understand how the agent and the host response are affected by spaceflight. And so in the first experiment, we looked at the bacteria, Streptococcus pneumoniae, a infectious agent that causes pneumonia. Droplet precautions are implemented in many of those cases. There is a vaccine against Streptococcus pneumoniae, but I'll explain that model organism is what it is in just a minute, a little bit more. The second experiment, I concerned myself with the host response. This was called Mouse Immunology-2. And it involved actually flying the mice themselves along with the space shuttle to the ISS for 16 days, for 2 weeks, approximately 2 weeks, where they were enclosed and exposed to microgravity, but lived in a contained space. And this is a small sample size because this is a spaceflight mission on a limited vehicle. The shuttle does not have infinite space, and so everyone has to really confine what they're studying to a small sample size, which makes publication more difficult. And so the culmination of these experiments, as these are really applied research studies, really the answers that we came up with helped build our knowledge of how our interaction with microorganisms really depends on the environment and on Earth and in spaceflight. That how we respond and how the microorganism or infectious agent responds is environmentally dependent. And that includes the environment of spaceflight, which carries significant risks of radiation exposure and a lack of gravity. And in order to tease these apart in these flight experiments, it can be difficult and potentially confounding. Radiation exposure is presumed in all of my experiments. We expect, and we, as biologists and microbiologists, understand the effect of radiation exposure, that there is a dose-dependent relationship with radiation exposure predisposing you to disease or DNA damage, particularly cancer. And so because this is ever present on the ISS and isn't something we could control in this experiment, we tried to make this more about the effects of the gravity sensing mechanisms within cells and within the host, how the disease after acquiring the infectious agent and the transmission of that disease, the virulence of the agent is changed by microgravity, by growth and cell division in microgravity. So using cells in a host, like the immune system, I particularly concerned myself with was the respiratory tract, because that is where most where the effect of gravity and the effect of microgravity likely be seen the most as an inhalation route. And the particular immune system around our respiratory tract is very different than our global immune system. It operates slightly differently. And the investigator that I worked with the immune part of this was concerned with, are the cells in the immune system, because they are continuously having to adapt constantly, unlike other parts of our body. Our other parts of our body don't necessarily differentiate into different cell types, express different genes, enhance cell-to-cell communication, or dampen cell-to-cell communication, other than the immune system. It is the cells of the immune system that do this the most in our body. They're continuously adapting to a threat to the health or safety of the host. And so they're really ideal for trying to understand how the host responds in a defined and measurable way. So part of microgravity is that in cell culture, which is how many bacteria grow, it is a low fluid shear environment. And I'll explain more about that in a minute, in particularly our ground-based models. Gravitropism is something I have not studied myself, but it has been observed that plants have a gravity sensing mechanism. Rock-like statoliths, structures within the cell that will sediment and then provide a sense of direction for the plant to grow away from the root. These types of experiments are still really, the mechanism behind this is still being understood, but it seems that plants really know how to grow away from their root. It is actually the fact that the plant can grow in different directions that alters the distribution of the hormone, the growth hormone in the plant. That's all I can say about the plants. And there has been repeated evidence that I'll go over later that there are gravity sensors within the cells of our immune system. Our experiments and other experiments have demonstrated that to a publishable level. The alterations in gene expression were overwhelmingly observed within bacteria. So in order to study bacterial gene expression in modeled microgravity or modeled spaceflight, you need to have a particular device. So we can fly these bacteria into space, but when we do that, we can get one set of data. If we want to prepare the experiment or try to guide our objectives and then replicate results, we need a ground-based model. And what was observed on the International Space Station in the 1990s was a large amount of biofilm production by the bacteria that had taken residence in the International Space Station. The International Space Station is in microgravity. It's about 250 miles above Earth. And so the lack of fluid shear, due to the lack of gravity forces, or the micro amount of gravity forces, that understanding allowed for the engineering of this instrument. It's a rotating wall vessel called a bioreactor. And the way that it works is, in order to model low sheared modeled microgravity, that's another NASA acronym, this is in this orientation. This is the control. So this is normal ground-based bacterial growth here on Earth. That's our, obviously, ground control. And this is how it really works, how the cells maintain in a fluid orbit. Because of the solid body rotation of the media, and due to additionally the sedimentation effect, the cells in this bioreactor really are, their lack of motility and cause to settle at the bottom of the vessel really allowed for continuous suspension of the cell in orbit. And so it's, the path of the cell is like this, and even as it goes down with some amount of sedimentation effect, it stays in an orbit. And with those experiments came quite a bit of data. The bioreactor really showed, and let me go back, really showed changes, global changes in an entire set of genes in multiple experiments, not just with Streptococcus, but with Staphylococcus aureus in particular. And that's one of the references I have. One of the other students in the same program that I was in was studying this. And it appeared that in this situation, a commensal organism, something that is on our skin all the time, Staphylococcus aureus, actually increases its attachment, its virulence, and its carotenoid production. But other microorganisms didn't. They maybe actually had a decrease in virulence. Salmonella, Burkholderia, we'll see the list of other organisms that were studied. And every organism actually reacted differently. And this was part of what made it difficult to pin down exactly what the effect of microgravity is on these individual bacterium. Some kind of migrated in their genes towards a phenotype or a type of being that is more commensal and less virulent. Actually, something that would have made us sick on Earth could be on us in space and not make us sick. And so these results were all variable. And not every model, ground-based model for spaceflight, is perfect. The bioreactor is not one. The biofilm that was noticed in the ISS was also noticed in these bioreactor experiments. And it's that low shear forces that allow the cells to eventually aggregate. So they're in orbit, but they eventually aggregate. And that is what a biofilm is. A biofilm is essentially a community, almost a superorganism, if you might want to get fancy with it, but a community of microorganisms, bacteria that decide to live together, communicate within each other, even if they're not in the same species. So they can be very dangerous on Earth. When they form in the ISS, what is their potential danger? They're essentially globs of bacteria. And it's not easy to predict that right now. But the other ground-based models for parabolic flight have provided us with some information to start with already, and for me to direct my research path. So there is the Vomit Comet, the parabolic flight of an airplane that, and really this is not a normal airplane that anyone is in. You can do this recreationally, the zero-G flights, although zero-G is not what you are in. But as the parabolic flight continues, there is a point where, and this is really aerobatics and not what a plane normally does. The plane rises in a steep axis and then drops in a very steep axis down. And that drop down gives a 3 to 5 minutes of essentially microgravity. Everyone in the plane is up and everybody's nauseous. And for looking at things that happen within a 3 to 5 minute window, reactions or activations of cells, not necessarily gene expression changes and virulence or any of that, but you can measure that by doing parabolic flight. And that there are multiple challenges with that model. It stresses live organisms out to do this over and over again. And so it isn't a good model for humans and how we would respond to that. It's a good model for how we would respond to stress, which is a continuously confounding factor when you're dealing with animal research. And we'll talk more about that with the mice. And so the clinostat is another type of bioreactor, really. It continuously rotates the sample in a way that is meant to mimic microgravity. You can use this in an assay model, like with plates, little wells, to see the actual sedimentation of the statoliths in plant cells. And this has also been used for immune cell culture experiments. These clinostat experiments are able to show dysregulation in cell signaling in the immune system, because that needs to happen quickly. The T cells have to divide rapidly. Antigen presentation and cell-to-cell communication need to happen quickly for the immune system to have a rapid response. And those molecules need to move with the circulatory system of the whole organism, actually, in order to get a proper immune response. And so you can see that gravity may alter this, and does alter this. The hind limb suspension model is what I used for my second experiment, and it has multiple confounding challenges. It's used for rodents primarily, really only. The human analog was being done at the University of Texas Medical Branch, where I left after finishing my degree in epidemiology. These experiments happened in conjunction with the University of Texas Medical Branch, Johnson Space Center, and Kennedy Space Center, all within the shuttle program working together. And part of this was a unloading bed rest study that was being done at the hospital. And these were called pillow knots. They were essentially lying in bed, but lying with their head pointing downwards and an axis. To see if I can do it. The head is pointing down. The feet are pointing up. The patients are like this, lying in bed. They have to, they're constantly having unloading from their legs and loading on their upper body. And this was showing the results of stress on the body, these studies. In fact, they were halted early because there was a apparent increased risk in retinopathy and higher intraocular pressures in the eye, because you are forcing all the fluids down into this area, and the inside of our eyes has a specific type of pressure that it needs to be under. And so these studies were halted early. And importantly, they really didn't, astronauts are not necessarily in that position. It wasn't a good model really at all. And much of what was found, the countermeasure was immediately identified, right? The hind limb unloading, or the unloading of the lower limbs in humans, can be counteracted by exercise. treadmill, right? Bone loss and atrophy of the muscle all can be counteracted by this. You can strap a person up and make them run. And so there's a countermeasure. So pillow knot experiments were not really that impressive. With the animals, in both Spagus-2, which is Streptococcus pneumoniae expression of genes in space, and this was the second experiment done in that category, and Mouse Immunology-2, we used two types of controls. So we had mice that were in our ground-based experimental vivarium cage control. So they're still on the ground while our other mice are in flight, and they have all the same lab conditions. They're in the same animal enclosure molecule with lighting changes for day and night and temperature regulation and diet. And as with the cells, the growth of the cells that we studied during Spagus-2, the bacterium, that there was a standard lab culture conditions, and there was our experimental apparatus on the ISS. And these were synchronous. They needed to be conducted at the same time as the flight experiments in order for all of the data to match up. So the subjects were in the mouse immunology group. There was another control synchronous group that was in a model of the space shuttle that NASA has at Kennedy Space Center that wobbles and goes back and forth and does all the yaws and movements that the shuttle vehicle does, so that they don't have the radiation and microgravity, but they have the launch and reentry stress, because that can confound our data. If we're looking at how the immune system is reacting, external stress in the environment will confound what we see. And we understand that, except part of that, right? But if you want to control the stress of the G-forces in launch and then the stress of the G-forces in reentry, that's one thing. But with the bacterium, we actually kept them fixed. And so they didn't start growing until they were in microgravity. Of course, the mice, we can't control that. They're in their cage, subjected to the same G-forces as the astronauts, only they're not strapped down. And when they re-enter, subjected to the same G-forces, but are not strapped down as well. So I think this is a duplicate slide. The basal control for the animal subjects and similarly for the cells are another control that we had to run. We had multiple controls that are equivalent to the flight subjects, but they are sacrificed at launch to obtain a baseline for this. So we had another group of mice. We didn't do this with the cells, bacterium, but we had another group of mice, of animal subjects that we had in the lab, same species, same clone, and just right at launch, they're sacrificed. And so we have a baseline of what a mouse right around launch looks like for comparison. And then this microgravity simulation, the Japanese Space Agency has picked up a lot of this research after Shuttle retired, and they found a way to model the microgravity simulation for, I'm sorry, I apologize, I'm speaking early. This model, the microgravity simulation model, is what we used for the rodent tail suspension for trying to simulate microgravity and repeat our experiments and see if we could reproduce our results on ground. And this was originally identified as a model for microgravity in vertebrates with a tail by Mori and Holton in 2005. So that is the microgravity simulation for the rodents. So it's similar to the pillow knots, except much more stressful. Much more stressful for the mice. And so here's the official NASA document that shows the first one, Space Transport Mission 118 with Dr. Niesel. And this was our model organism, Streptococcus pneumoniae, that launched, the initial one launched in 2007, and the one I was a part of is STS-129, Spegus-2. He is our principal investigator, Dr. Niesel again. And their launch date was right before Thanksgiving, and in a good time to fly in Florida. This was the crew. They were not our subjects. We'll get to the subjects in a minute, but the crew was extremely important because for this experiment where we were sending up bacterial cell cultures to space, the astronauts were actually the ones that conducted the experiment for us. On the ground, we did the simulation controls, but the actual experiment was conducted by likely an engineer, somebody who's potentially not a microbiologist at all. This was the launch. This is about as close as you can get, being part of the mission, while not being all out on the launch pad. And this is Space Shuttle Atlantis 129 leaving. So let's talk a little bit more about our subject, our research subject. In liquid culture, we had Streptococcus pneumoniae shown here. It is a typical bacterium that can just be a colonized organism on our body. But when it makes or overgrows or makes trouble, we can have severe disease and even death. And so you see, as a human, we have an oral airway and an oral opening to our gastrointestinal tract, along with an oral airway. And that's part of what makes being a human dangerous is, you can have colonized Streptococcus or even a strep sinus infection. And you can go a couple of different ways. You can get sick and start shedding it, and then it drains into the eustachian tubes connected to the sinuses and causes otitis media, which is an inner ear infection. The bacterium can be transmitted to other people while you're sick with this via droplets. And it's very common. This is within many of us. And once you get to the invasion stage, what can happen is, like I said, we only have one opening here. And so if here in our sinuses we are shedding and have an overgrowth of Streptococcus pneumoniae or an overgrowth of a strain or a serotype of this bacterium that is more virulent because it mutates quite a bit. And it, instead of being able to cough it up, it goes down into the airway and to the lower parts of the lung where gas exchange happens. And this is pneumonia. The gas exchange in the lungs needs to happen here, but instead it's filled with fluid. And that's pneumonia. Additionally, it can then move out of the pulmonary part of our body, the pulmonary circuit, into the circulatory system where the blood circulates. And we then can get septic, bacteremia, and it can go to the brain and cause meningitis. So it is an organism worth studying because it does carry a burden of disease in many populations. And so in order to do this experiment, what we had to do was use special devices. So this was the objective. The objective was to look at the growth and the gene expression changes, protein production, and virulence, overall virulence of strep pneumo that grew only during a space mission. These were up there for additionally approximately 2 weeks. And this was 18 days. What happens is the apparatus, the fluid processing apparatus, has these separate chambers, and they're packaged together based on what each experiment is supposed to be. And the idea is that there is a rubber septa, so this fluid processing apparatus, are the glass-filled tubes, and these are the rubber septa that separate the different compartments. The bacterial sample that's just bacteria we grew up on Earth in there, and then the culture medium that the bacteria needs to grow in, because bacteria doesn't grow without a culture medium. It's in here, freshly entered in by me and my colleagues. And then this septa filled with the culture medium that the bacteria needed to actually grow. And so the astronauts, once they were at microgravity, were to dial along and push down the rubber septa. In this one model, it's just red. Bacteria into culture for growth for that duration of time and space. And then at different time points, different fluid processing apparatuses would be activated fully down to this yellow chamber. And this yellow chamber, media, inoculum, and then processing agent. So this is the inoculum, the media that go together in order for the actual microorganism to grow. And the processing agent was something that was meant to fix the cells exactly as they were in microgravity at that time point. So the cells have grown for maybe 3 or 5 days, and now we are going to fix their genes by pushing this all the way down so that the genes and the expression of those genes, the amount of protein, all of that was essentially frozen in time and fixed. And so when we would get it back, the idea is that we would take the summation of these 3 things, essentially bacteria that have been allowed to adapt to spaceflight microgravity, and then fixed in microgravity. So no re-entry stress, no launch stress, none of that, just microgravity. And then identify the genes that were different. And additionally, inoculate mice with this infectious material, because not all were fully activated, right? We allowed for some of these gaps not to be activated. Some chambers didn't have this inoculation medium, this processing agent in order to fix the cells. We allowed many of these to grow and grow and grow and grow and grow in space. And then they were subject to the re-entry stress, but they're bacteria. They divide often, adapt. And so immediately upon landing, in order to get this pathogen and examine its virulence, we injected the inoculum, the bacterium, into 333 mice. I wanted to show this here so you could see that the hardware that is used on the ISS. Bioserve was our main company that we worked with. They provided the test tubes, the fluid processing apparatus. This is an AEM, an animal enclosure module, where the mice were kept when they were in spaceflight, and different types of instruments, tissue preservation kits that are all designed to work on the ISS and really provide you with the best, it's industry meant for studying cell and human research in space. And so our subject, our ground-based subject, was the bulb sea mouse for the bacterial inoculation. The C57 black 6 mouse, I'll talk about later, that group of mice was inoculated with virus after they had spent time in space. Our mice, these little pieces of popcorn right here that are very kind and sweet, they don't even really bite very much, that was our subject to inoculate and do the virulence challenge on. And so we don't sacrifice these mice. We only see how quickly they die, or how sick they get before they die. And we chose these 300 mice, all clones, the bulb sea mouse, because it has a moderate tolerance to radioactivity. And so the effect of radiation will not necessarily be affected. I suppose if we were going to send them to space and expose them to radiation, that would be more important. But what's actually even more important was their immune phenotype, the way their immune system works. They have, it's very easy for them, these particular mice, to activate the innate part of the immune system. They're skewed in one direction. They have high complement activity, so they have a rapid response to bacterial infections. And then they help with these particular mice also have particular susceptibility to different types of bacterial infections. And all of that, when you're trying to examine how the host is reacting to something, and you know your infectious agent is a bacterium, that guides the model organism that you will use to test that on. They have a low incidence of cancer and carcinogens, so they are more reactive to certain bacterial agents, but have a very good initial inflammatory response. And so their immune system should immediately activate. They're not in any way immunosuppressed. When it came to our gene expression data, we lost quite a bit. We were able to identify 213 genes that were changed in expression in the Streptococcus pneumoniae. We did this through mRNA, microRNA analysis, because when the processing agent is added to the bacterial culture, it fixes the mRNA, the actual expression of genes before they've become proteins. And what happened is, this is the way that this is set up, and the astronaut was given specific instructions. And unfortunately, through many of these, the fluid processing apparatus, instead of being inoculated and then fixed, many of them were inoculated and then fixed at the same time. And so what we had was essentially bacteria frozen just after a few seconds of being exposed to the culture medium that they grow in. So what we did find, though, are those 213 individual genes, and they were significantly altered in result to spaceflight. We were able to establish that. But the group of genes and how they affected the actual bacterium, that part of the research process and data analysis has actually not still been completed. Because with NASA grants for spaceflight experiments, the money is given for the experiment and not the data analysis process. And the principal investigator, David Niesel, and all our collaborators that did eventually publish, they published modeled microgravity experiments before this, but were not able to publish after, because it is really the culmination of everyone's results. What we found is a collective amount of evidence that microgravity increases the virulence of Streptococcus pneumoniae and enhances the virulence of bacterial growth in space, specifically with Streptococcus pneumoniae. And we would need to devise countermeasures in order for long-term spaceflight habitation. What happened was, we injected these 330 mice. Well, no, we divided them. They were control groups. There was the ones that just got phosphate buffered saline, so water. And there was the group that was inoculated with 10 microliters of bacteria from space, intraperitoneally. And it was rapid. The motion is grab the mouse, pull its head back so it doesn't bite you, inject. Throw it down, pick another, inject. We had to do it all within one. We made it to like 41 minutes. Had to do it all right way before the bacteria has a chance to change its expression and adapt back to be on Earth. So we inoculated all these mice, put them in the cage. The mice that received the Streptococcus that had flown on the ISS lost weight faster. So the way that we measure virulence and disease in these mice is by weight loss, lethargy, and then eventually death. The non-control group, right, the experimental group, Not only did all of them die, whereas the control group, not all of the mice died. And I apologize for not having this data in front of you. It's actually on a computer that I don't have access to right now, and I actually hope to get access to through the resources of this college, where I actually have some of the graphs. But essentially, these mice got sicker faster that had the space bacteria. They lost weight faster. They became more lethargic faster, and then all of them died. And typically, this microorganism, when it's given intraperitoneally, the dose and amount of colony-forming units, which is how you measure the amount of bacteria you're giving them, it's not fatal 100% of the time. There's usually disease, and then weight loss, lethargy, potential recovery. Some mice do die, but not all. And so this was an obvious change in virulence. And the mechanisms behind it, the gene expression, is really what was left out of this experiment. Which genes are associated with this virulence in Streptococcus pneumoniae that's increased during growth in spaceflight. So the next experiment, I needed to concern myself with the host, and this was Mouse Immunology-2. Dr. Roberto Garofalo actually participated in 2 of these. I only participated in 131. In 135, he collected spleens from a different mouse and looked at alterations in interferon production in a different group of mice. But for Mouse Immunology-2, this experiment was really to discover what triggers and leads to increased susceptibility of infection in the respiratory tract. The idea is that the overarching hypothesis was that there's a combination of several factors, from microgravity to stress, the actual stress of flight on a host organism, that will affect, and we're extrapolating here because we sent mice, the human immune system during spaceflight, and that will result in impaired antiviral response in the respiratory tract. We knew there was an impaired antiviral response already in spaceflight because there had been reactivation of all herpes viruses in space, including the one that causes chickenpox. And so we knew viral latency was affected, the ability of a virus to kind of go dormant for a while and not cause disease. Specifically, we wanted to look at the role of the microgravity and stress in relation to infection with respiratory syncytial virus. Respiratory syncytial virus is a virus that Moderna is currently working on a vaccine for. It causes a significant burden of disease in young children. RSV is a dangerous virus for young children to get. The effects on trafficking and function of the specific cells that migrate back and forth and patrol the respiratory mucosal membranes were also to be looked at. This translates to the ability of the innate immune system to communicate to the adaptive immune system. Interferon and in the respiratory tract helps with the T cell mediated immune response. And so we have our inflammatory innate response, and we have our T and B cell immune response that has memory and effector cells that can kill virally infected cells, and then remember later. And then that is the premise behind vaccination. I have memory. So these cells are very important in the respiratory tract. So we looked at type 1 interferon because it is really the stimulator of antiviral genes that are targeted against preventing virus replication within the cells that it goes into. So we wanted to look at the production of this molecule and see how effective it is, and whether or not virus infection can alter the type 1 interferon, which is essentially the communicator between the innate and adaptive immune system, in a different fashion, autocrine, paracrine, affecting the cell itself, or affecting other cells around, producing this interferon can actually go back on the cell itself and reactivate it. So this is a dynamic and much more technical part of this talk. And it's important to have type 1 interferon that is proper because it regulates the immune response. It regulates the inflammatory and adaptive immune response. Many patients that died of on ventilators with SARS-2 actually died because their bodies made an antibody specifically to interferon. And so if you have an antibody against your own immune system's communication molecule, there is no antiviral response and there's no beating the virus. And that's one of the things we've learned about this viral agents, how important a regulated immune response is. And so we chose the C57 black 6 mice, 16 of them. Now, these are much more aggressive mice, but they also have a very high interferon production. So you're guaranteed to get some amount of interferon production. They're similar in many ways to the BALB/C mice, except that C57 black 6 mice, the BALB/C and the C57 black 6 mice, they skew the T cell response, right, to a type 2 T helper cells response and a T helper 1 cell response. And what you really need to know about that is the T helper 1 immune response and interferon gamma production play the key role in the response to infection in these black 6 mice. And so there were an ideal subject. Alternatively, it's easier to induce a Th2 response in the little white popcorn mice. They are more common to have allergic reactions and infectious diseases. So you see, we wanted a mouse that would produce enough of what we wanted to study in the cells that we wanted to study. Whereas the first group of mice, we really wanted mice that would get sick, would definitely get sick. So you design your objectives using your model organism to be able to get what you need, the information you need to get. These mice are commercially available for these specific reasons. That's how one studies a complicated biological systems, really by knocking out genes and seeing what happens when certain genes are present or more active and versus when they are not. And so we were essentially trying to identify the vulnerabilities that spaceflight introduces to the respiratory immune system. These mice were also enclosed in modules like this. You see, they were protected from the astronauts even. The astronauts could not give them any bacteria. They were in a closed loop containment glove box and in a module that's essentially a drawer. They have water, they have access to water and fans. There's blowers. It isn't an animal enclosure module. It's not great, but it can support 10 mice. And there's a continuous airflow to remove the otherwise would be ambiently floating waste from these animals. This mission got scrubbed 4 times, and so it made it quite a bit more stressful. The co-investigator, Theodore Van Cook, still studies this virus, but her and Roberto Garofalo is now studying more SARS-2 and RSV. And so this research, again, was done, findings were made, and later was picked up by another group, which I'll get to follow up on. This is a great picture because this was STS-133, and it was the last flight of this space shuttle. This was in 2011, right before the mission was ending. And it got scrubbed multiple times. And so the thing that's frustrating when you're working with vertebrates, can be frustrating, is you have to deal with several committees that make sure what you're doing is humane. And that's not to say that we want to do a bunch of inhumane things to these animals. It is just that every time we load our 16 mice onto this shuttle and it moves, it gets dragged out to the launching pad and sits there, and oh, there's a couple clouds in the sky. We're scrubbing the launch. This thing gets dragged back over there and the mice get sacrificed because they're not ready for the experiment. They've been through the stress of being in this thing and moving all around. And so we lost a lot of mice. It was difficult. It was difficult to do that. Inoculating the mice when they returned with RSV after 14 days aboard the ISS was immediate. So when the mice returned, the first thing we noticed is they were moving around on the ground using their chins and not their arms and legs. They had gone through significant muscular atrophy during that time. And so they were flat like pancakes and looked extremely sick and weak. And so the principal investigator and co-investigator decided to give 8 mice to change the dose of RSV that we were going to inoculate them through the intranasal route. It was reduced at the last minute. It was going to be 10 to the 7th particle forming units, which is how we measure metric for viruses, particle forming units, 10 to the 7th, right? At the last minute, the principal investigator lowered that. He said, drop it by half a log. These mice won't, none of these are going to survive the 3 and 5 days that we need to sacrifice them post-flight. They're all too sick. And so we dropped the dose, half a log, to 5 times 10 to the 6 particle forming units. And the other 8 mice were given control, phosphate buffered saline, through the same route. And then they were sacrificed at days 3, 5, and 7. And we actually had an additional set of mice, I haven't listed it here, unfortunately, that we inoculated with the same dose of 5 times 10 to the 6 particle forming units while they had stayed on ground and been through that pretend shuttle that moves around, rocks them around, and stresses them out. And so what happened, likely, the reason we didn't get really any publishable results and other data that was just too inconsistent among the subjects was likely due to the drop in dose. No mice died from viral infection. We sacrificed them in order to retrieve the sample that we needed to study. And so I'm going to move on to that. And I just want to warn you, if you're squeamish, you may not want to look at this slide. But we had to collect the signaling molecules and the cells that migrate and live in the respiratory mucosal tract. And the procedure for doing that is a bronchoalveolar lavage. In humans, this is done by a physician in a hospital, but a nurse got to do it on a mouse in this experiment. And I was excellent at this procedure because I'm familiar with the instruments used. This is the protocol for a mouse bronchoalveolar lavage. So they are sacrificed because we don't want to disturb anything in the airway. We don't use, we don't gas them with CO2 or isoflurane. We give them an injection of essentially morphine, and then they go to sleep and we cut their femoral. And so they essentially exsanguinate. And then when the mouse is asleep and deceased, an incision is made under the throat. You use a catheter, a particular size catheter, and a small syringe, a tuberculin syringe, because you really only get out maybe 1.5 to 2 of fluid out of their lungs. And so you pull open with forceps the outer area surrounding the trachea, expose the trachea, and make a small incision. The incision then, as the mouse lies flat, is open and ready for the insertion of this catheter. The catheter is filled with a certain amount of saline, and it's a lavage. So the mouse lung only holds about 1.5 to 2 mLs. And so we inject first 1 to 1.5 mLs of, and milliliters is what I mean, of saline, isotonic fluid that will suspend all of these cells that are in here and chemical messengers that are in the lungs reacting to virus infection after spaceflight, and then go through this centrifuging procedure and creating a pellet and going through more centrifuging procedures and lysing red blood cells in order to get that out and stopping the lysis. All a nice recipe. And at the end, you should end up with cells that you can count on a hemocytometer or look at under a microscope, or send for RNA and protein extraction, lyse and then send for RNA and protein extraction. And so we did do that. And of our 8 mice that we sacrificed, there was so much inconsistency between each subject's reaction. The only way we could describe it was complete immune dysregulation in the upper respiratory tract of these mice. That we don't know how long it would have lasted because we kept the mice, we managed to keep them alive for the 7 days. We didn't give them lethal doses, and that wasn't what we were trying to do. By dropping the dose of virus that these animals got, we were not able to get a clear picture of what true infection post-flight would have looked like. And so instead, the cells didn't have a real and tremendously robust response, but the dysregulation between the activated cells, the cells that were communicating with each other, the amount of interferon was inconsistent with each subject. And so we had to do ground-based reproducing studies. And this is what I spent many years doing. This is the hind limb suspension model. And it actually was more tape and then tape up here, but then a loop so that they can move along a track while their hind limbs are raised. And this is supposedly a method of modeled microgravity. And so I spent quite some time inoculating mice with different amounts of virus and stringing them up. And just in this process, a night of this is often too stressful for the mouse. The mouse will just die, even if it's a control mouse. And then when they're inoculated and have the virus and they have illness and they're tilted down, they're able to shed out, mucus and out of it. It's somewhat helpful to be laying downward, right? This mouse, what is this? This is a mammal that's trying to fly, right? And so things get out, and that's okay. So there were multiple confounding things that really weren't able to give us anything. And so I worked on it as much as I could. We weren't able to gather really anything except poor cell-to-cell communication and dysregulation of the immune response to a virus. But others, the Japanese Space Agency picked up this research and really wanted to understand how spaceflight not affects the innate, but the acquired immune system, the cell-to-cell communication, the chemical messenger part, and the generation of the cells in the adaptive immune system. And what they found is the site of education and growth of T cells is the thymus. And mice that lived on the ISS in microgravity had reduced proliferation of immature thymocytes, which are immature T cells. And so there was just a simple reduced amount, an atrophy, and reduced cell proliferation of a very important part of the immune system, the T cells, in mice that were in the ISS. So their T cells were diminished, the amount, which would cause a poor immune response to a viral infection. Additionally, what was found is the origin of all of the immune system cells, many other cells, is really the bone marrow. And so here in the bone marrow, what happened was the stem cell, the mesenchymal stem cell that can differentiate into all of these things, bone cells, cartilage cells, muscle cells, fat cells, that the chemical messengers here, CXCL2 and the, so this is a cytokine here, which is a chemical messenger, and this is an adhesion molecule for this cell to be able to attach to something and be part of the endothelium. The function and differentiation, which is really what's key with a stem cell, because the point of a stem cell is that it can differentiate into different types of cells. That's why they're so great. There was a difference in their homeostatic condition. There was a change in the bone marrow in these mice, and it changed the distribution of these types of cells, which may not play much of a role in the acquired response exactly, but demonstrates that these chemical messengers and cytokines were not maintaining a homeostatic environment. And so because of that, differentiation and proliferation is going to be altered. There's going to be altered differentiation in mitotic divisions of these stem cells because of microgravity and space radiation combined. And when you want to separate that out, this group was able to do some of that. Space radiation really seemed to have more of an effect, and a bigger question there is the mechanism, and on fat cells, almost like the body is adapting to be blubberous, right? And a diminished generation of bone, almost like when we're trying to get lighter bones or adapt. And because of the diminishment, the diminished activity of this chemical messenger, CXCL2, that resulted, and we know this is more to do with microgravity, the shape of the internal skeleton of the cell that is made of a protein called actin. There were changes in this proliferation and differentiation in the activity of these internal cell signaling pathways that then led to further differentiation away from bone to an adipocyte. I'm not going to get into all of the potential inflammatory and health detriments of having too many adipocytes, which are fat cells, versus bone cells. But what you can see is almost an adaptation happening in the stem cell, right? This adipose tissue is lighter than bone. We're able to float and move around in space if we're more buoyant, more light. And this was where these investigators really looked at what we had already observed as far as the space stress and reactivation of human herpes virus 1, particularly in regards to the CD8 killer T cell. Because if this virus is latent in the trigeminal nerve, as it is, this is the oral herpes virus, and it is reactivated due to the stress of space, which could even just be radiation, right? Ionizing radiation causes DNA damage, which makes the latent virus concerned about the safety of its genome. It's hiding in this neuron back here. Reactivation caused by stress is because the virus is trying to jump. It's trying to leave this stressed out host and go somewhere else. It doesn't want its DNA damaged. And the T cell that is supposed to take care of this virally infected cell, is dysfunctional. These cells were completely dysfunctional. So cells already infected with a virus we don't properly make functional T cells in order to eliminate the cell that has the virus, that is producing virus. And that's the point of the CD8 T cell. A functional one would instead, when we have latent infection, a functional CD8 T cell would go through this intracellular pathway and not allow for the transcription of viral genes. See, this ICP4 leads to transcription, which is expression of a viral gene. Instead, with a functional CD8 cell, as identified in a virus, that is blocked, and so there is no expression of viral genes, and so there is no reproduction of the virus. There's none of that. And so this brings me to really the end, which was my final question, my original question of like this 3 to 6 meters or 3 to 6 feet or 1 to 2 meters decision. And it hasn't been solved in spaceflight, but because of the pandemic, it was reevaluated. Airborne or droplet precautions for healthcare workers treating coronavirus disease patients. And their findings make perfect sense and really align with the idea that certainly many more infectious agents will become airborne in a contained spaceflight vehicle. Because if you look at, and so here is the actual key, modeling, experimental studies, and human studies, right? So there's a limited amount of human studies. But in all of these modeled experiments, a droplet latent, turbulent with bacteria droplet of approximately 30 micrometers in diameter. This is the droplet, right? 2 to 3, 1 to 2 to 3 meters. Right? Droplet is here. So that's on regular gravity. And then in more modeled experiments, a full spectrum of droplet sizes was identified. So we're not talking about aerosols. We're talking about droplet sizes that have different size, weight, volume, because each infectious agent has a different size, weight, and volume. And it looks like when you look at the full spectrum of them, it can go out at least to 26 feet, 8 meters. And so by reevaluating this, the maximum distance recorded in the few available studies of this says it's more like 26 feet, depending on the size. So depending on the amount that gravity would really affect when that infectious droplet would hit the ground. And this study really emphasized in its discussion that this prescribed approximately 1 meter, 3 feet, even really 6 feet, that are associated recommendations for healthcare workers in dealing with infectious agents at ports of entry, like the inhalation route, are not and were never based off current scientific evidence. They were based off of scientific evidence that the first group of epidemiologists who were ever studying these ideas came up with, and they were stuck with for a very long time. And to try to understand them, transmissibility, and the effect of microgravity indirectly was this. It was trying to identify if something can become, depending on the environment, more virulent, more transmissible, or less virulent, less transmissible. And it does depend on the environment, and that includes microgravity. These are my references, and that is, oh, did I mess everything up? That's the end of this talk. I think I went over quite a bit, didn't I, Erin? I don't think I can hear you, Aaron.
We actually had a question in the chat for you from
great.
Caleb, who had actually asked back early in the talk when you were talking about the creation of biofilm on the space station. They asked what were some of the possible outcomes if the biofilm culture had come in contact with the human astronauts on the space station.
Yeah, that's a great question. So you can look at old publications from Mir and Space Lab that preceded the International Space Station. Mir was cultured and found to have tremendous amounts of biofilm. The implication for their health is that these globules of potentially diverse and interacting microorganisms that adapt pretty well to microgravity could enter through the inhalation route or even enter through contact with an object, an inanimate object, the fomite route. And that these biofilms would accumulate under fingernails, accumulate in spaces on our body that are not meant to have an overgrowth of whatever bacteria is growing on the ISS. That has not played out to be a big risk because of continuous cleaning and what they've learned from Mir. And but biofilms have a very significant, they contribute to disease here on Earth greatly. They accumulate on mechanical devices that are implanted, like joints, arthritis, replacing hips, that type of thing. And that causes disease. It's difficult for bacteria to penetrate biofilm. They're kind of a super community. And we learned from this more about how biofilm production works and how we can combat it and use countermeasures against it. And so while there was a potential risk to the astronauts, what we actually gained from this helped human health on Earth and helped develop a countermeasure in space. Very good question.
It was a good question from the audience. I actually had one that came up because I had flashbacks for a minute. When I was doing my undergrad work, we did an experiment that went on the KC-135 doing the parabolic orbits, and I was just curious because I know when they do it for the human experiments or for the larger experiments that we were doing they were doing like 20-second parabolas. So you only got like 20 seconds of microgravity. How did they manage to do it for longer than that for some of the other ones?
You know, the 3 to 5 minutes is isn't just the microgravity, right? So it's the kind of the effect on the whole organism. And so you're right. There's multiple problems with this model. You truly only get 20 to 30 seconds of true microgravity. There was a push for suborbital flights that would have given longer amounts of microgravity. Blue Origin actually was one of them, and the Virgin Atlantic is part of that suborbital flight. And not for recreation, but for scientific exploration, to run experiments where you need microgravity for potentially maybe an hour, however long everyone up there can tolerate that suborbital flight, which still has not happened.
Okay,
Did you get to go on the KC-135?
No, I got to go in it before it took off, but with all my medical conditions, they weren't about to actually let me go airborne. That was not going to happen.
No. It's I haven't actually done it either myself because I have a weak stomach, But I went through all the other training at the neutral buoyancy lab with the hypobaric and hypobaric chambers that the aerospace medicine residents had to go through. And many of them went on the vomit comment, the KC-135. I don't need to do that. I don't need to vomit everywhere. Thank you so much for the questions from the audience.
We know it has its name for a particular reason. Everyone else can go look it up on YouTube if they need proof.
It still runs. I think there's one in Las Vegas you can go and get sick on.
Well, that's the last of the questions from the audience. So I think we'll take this time to thank you once again, Zinzi, for giving this talk. And we appreciate it. As I said, it was a very informative talk, and we look forward to seeing more of the results from these types of studies in the future.
Me too.
And with that, I will thank you all for attending today's talk, and hopefully you'll be able to join us on June 3rd for our final physics and astronomy lecture of the year, when we're joined by the director of the High Altitude Observatory in Boulder, Colorado, Dr. Holly Gilbert, who'll be talking about a new era in solar observations. Students, I think, would benefit from choosing SWOCC because it gives them an opportunity to get involved at a more hands-on level earlier in their career. Students choosing to come to SWOCC benefit from having opportunities at the freshman and sophomore level to get involved in research that can carry them forward in their career while still maintaining the same academic rigor as they would experience at a four-year institution. The day in the classroom in physics and engineering starts out as sort of a conversational approach to the material that we're looking at, whether it be solving rocket science problems, whether it be designing bridges, whether it be designing electrical circuit systems. It's usually a conversational approach that leads to a lot of questions. I always tell students if they're doing science well, they'll always leave science class with more questions than they got answers to begin with. And that's to drive the intellectual curiosity of the students. And that's what I try to do, making things interactive as possible and letting them foster that curiosity and develop those critical thinking and teamwork skills that they need to succeed as they go forward. You're dealing with a class in physics that's typically between 10 and 20 students. That gives you plenty of time to interact, ask questions. You get more direct responses from the instructor. And SWOCC being a small enough school, there are instructors that you will see a number of times, so you get to develop sort of a rapport with your instructors and truly do become more like mentors than they do become teachers. The biggest thing it brings is a sense of opportunity. It gives us a chance to revisit how our curriculum works. It gives us a chance to incorporate new ideas, new equipment, and it also provides the students with ample opportunity for new projects and new developments in terms of research. The physics and engineering programs at Southwestern are a great way to begin your journey in a professional STEM career.